Hi, my name is Kyber Savage and I'm doing Biology AS level. And in this video I'm going to be looking at my least favourite thing in the whole of this topic, which is the immune system and the immune responses. So to begin with, the skin and the mucous membranes are the body's primary defences. The skin acts as a physical barrier, blocking pathogens from entering the body. It also is a slightly chemical barrier, because it produces chemicals that are antimicrobial. They can lower the pH and inhibiting the growth of pathogens on your skin. The mucous membranes, they protect body openings for exposing environments like your mouth, nose, ears, stuff. And some of these membranes secrete mucus, which is a sticky substance which traps pathogens and contains antimicrobial enzymes. But if a pathogen gets past this immune system response, God forbid, and the immune system will respond. And the definition of an immune response, if they ask, is the body's reaction to a foreign antigen. And antigens are molecules like polyproteins or polysaccharides, and they're found on the surface of cells. And when a pathogen, like a bacterium, invades the body, the antigens on its cell surface are identified as foreign, which activates cells in the immune system. And there are four main stages of the immune response. Number 1, phagocytes engulf pathogens. And a phagocyte, e.g. a macrophage, is a type of white blood cell that carries out phagocytosis, which is like engulfing the pathogen. And they're found in the blood and in tissues, and they're the first cells to respond to a pathogen inside the body. And they work by, first of all, they recognise the antigens on the pathogen. So phagocyte recognises antigens, or phagocytes on pathogen. Phagocyte recognises antigens on pathogen. Yeah. Then the cytoplasm of the phagocyte moves around the pathogen, engulfing it. Now the pathogen is contained in a phagocytic vacuole, like a bubble, in the cytoplasm of the phagocyte. A lysosome, which is an organelle that contains digestive enzymes, that fuses with a phagocytic vacuole, and the enzymes in that break down the pathogen. The pathogen then presents the phagocyte, lots of P's. The phagocyte then presents the pathogen's antigens. It puts the antigens on its surface to activate other immune system cells. So if they got it on the surface, other ones can come up and notice it and stuff. The second part in the immune system response is the phagocyte activation of the T lymphocytes. The T lymphocytes, just easy to remember, is another type of white blood cell, and their surface are covered with receptors looking like that. The receptors bind to antigen presented by the phagocytes. So remember when I said the phagocytes present the antigens on the surface, like sticking them to the surface? And that means that they can recognise them and bind to them. And each T lymphocyte has a different receptor on its surface. And when the receptor on the surface of the T lymphocyte meets a complementary antigen, it binds to it. So each T lymphocyte will bind to a different antigen. This activates the T lymphocyte, and it divides and differentiates into different types of T lymphocytes, and they carry out different functions. They either become memory cells. Memory cells are, let me just check on the next page so I can get this completely right. Here. Memory cells remain in the body for a long time, and they remember the specific antigen and will recognise it a second time round. So that's good. Also, some activated T lymphocytes release substances to activate B lymphocytes, and these attach to antigens on a pathogen and kill the cell. Yay! Okay, the third stage in the immune response is T lymphocytes activating B lymphocytes. That's what I said. And they divide into plasma cells. Woohoo! B lymphocytes are another type of white blood cell. They're covered with proteins called antibodies. These antibodies bind to antigens to form an antigen-antibody complex. Each B lymphocyte has a different shaped antibody on its surface, and when the antibody on the surface of the B lymphocyte meets a complementary shaped antigen, it binds to it. So each B lymphocyte will bind to a different antigen. This, together with substances released from the T lymphocyte, activates the B lymphocyte, and therefore the B lymphocyte divides by mitosis into plasma cells and memory cells. And then finally, the fourth final one, which are plasma cells, make more antibodies to a specific antigen. Plasma cells are clones of the B lymphocyte. They're identical to those last B lymphocyte, and they secrete loads of the antibody specific to the antigen in the blood. And these antibodies will bind to the antigens on the surface of the pathogen to form lots and lots and lots of antigen-antibody complexes. Sorry, deciding whether I should look at the structure of an antibody, but I will. Antibodies, it's quite hard to see, I'll show you in my book. Look like this. So the structure of the antibody means that there are variable regions of the antibody which form the antigen binding site, and variable regions are the little orange bits. Okay. Then the shape of these variable regions are complementary to a particular antigen, and they differ between all different types of antibodies. Then there's the hinge region, which is just a little bit where my finger is there. There's the hinge region. And that allows flexibility when the antibody binds the antigen. The constant regions, which are the little lines that come down, these are the two little constant regions, 1, 2. They allow binding to receptors on immune system cells, like phagocytes. And the constant region is always the same in all antibodies, which is easy to remember because it's called the constant region. And the disulfide bridge, which is in the middle, you probably have seen that lots of times now. That's the disulfide bridge there. That holds the polypeptide chains together. And antibodies are really helpful because they help to clear the infection in a number of ways. They agglutinate pathogens. This happens because each antibody has two binding sites. And so an antibody can bind to two pathogens at the same time. And this means that the pathogens become clumped together. And phagocytes then bind to the antibodies and phagocytose a lot of pathogens all at once. Neutralizing toxins, they also do. Antibodies can bind to the toxins produced by pathogens. This prevents the toxins from affecting human cells, so the toxins are neutralized, and the toxin-antibody complexes are also phagocytosed. And finally, they prevent the pathogen from binding to human cells, because when antibodies bind to the antigens on pathogens, they might block the cell surface receptors, and that means that the pathogens need to bind to the host cells, and that means the pathogens can't attach or affect the host cells. So, all very good. But the primary response is quite slow. The primary response is when an antigen enters the body for the first time and activates the immune system. So something completely new comes into your body. That's the primary response. But it's slow. Because there aren't many B lymphocytes that can make the antibody needed to bind it. They haven't seen it before. So eventually the body will produce enough of the right antibody to overcome the infection. But meanwhile the infected person will show all their poor symptoms of the disease. But after being exposed to this antigen, both T and B lymphocytes produce memory cells, and they remain in the body for ages. Memory T lymphocytes remember the specific antigen and will recognise it for second time round, and memory B lymphocytes record specific antibodies needed to bind to the antigen. The person is now immune, and they have the ability to respond quickly to a second infection. And I'll just go over the last bit about the secondary response, and that's fast, which is good. Because if the same pathogen comes in again, the immune system will produce a quicker, stronger immune response, because they remember it. This is a secondary response, and it happens quickly, because the memory B lymphocytes just quickly divide into plasma cells, then produce the right antibodies to the antigen. Memory T lymphocytes divide into the correct type of T lymphocytes to kill the cell, and the secondary response often gets rid of the pathogen before you show any symptoms. So if a cold that you've had before comes into your body and they recognise it, it will be gone before you get ill. So good luck with your biology AS level exam. It's coming soon. Revise!